Now it's time to talk about two last methods that biologists use to study organisms that are biotechnology focused. So the techniques to clone whole organisms and also how biologists use stem cells. So biologists engage in what's called organismal cloning, which is distinct from bacterial cloning, which I talked about early on, as a method for understanding that condition of totipotency that exists early in an organism's development. So, this helps to address a question of, all right, so we know that at the initial zygote stage, a cell contains all of the genes that it needs to develop into a full adult organism. But, do all cells retain that same set of genes? Or are there some cells that actually lose genes over the course of differentiation? So the way to get to the bottom of that is to take a nucleus from a mature cell and then transplant it into an embryonic cell, so a cell early in development, and then observe the developmental outcome. So that's just illustrated here for frogs. So if you take a less differentiated cell in a frog, you pull out its nucleus, and then you take a donor nucleus from a differentiated cell. So here, for example, a cell in the intestine, transplant that into the host embryo cell, and then wait for development to start. Then if you do that with an egg cell, you'll wind up getting an adult tadpole. In contrast, if you take the nucleus from one of these less differentiated cells and transplant that into one of these intestinal cells, you won't wind up getting a fully developed tadpole. So that tells us something about past a certain stage of development. Once cells have differentiated, there's something in the cytoplasm that is contributing to that differentiation that can't be reversed. And in contrast, it also tells us that the information that's in the nucleus remains consistent in those differentiated cells. So they're differentiated due to factors in the cytoplasm, not due to factors in the nucleus. And that also kind of reinforces the general message that as cells differentiate, they lose this ability to be totipotent, and that's due to changes in things in the cytoplasm. So the first animal to be cloned was Dolly the sheep. So she was cloned in 1996. And so this is the first time that we're actually able to take a nucleus from a differentiated cell and get it to develop back into a full adult animal. So there's been a lot involved in just figuring out the logistics of transplanting nuclei. And there are still ongoing studies with a lot of totipotent embryonic stem cell lines, again, to just try to have a better understanding of how different groups of interacting genes lead to differentiation into different major cell types. So across these lectures, I hope that you've gotten a sense that biologists now have a really sophisticated set of techniques that allow us to study a wide range of processes on the cellular and molecular level. These are things that I think if you talk to a biologist, from like the 50s or the 60s or the 70s, I think they'd be insanely jealous of what we can accomplish with the molecular tools that are now available. I mean, even a biologist from the 80s. But I hope you've also seen that these tools also raise a lot of different kinds of ethical questions. And it isn't as though deploying these methodologies has allowed us to solve biology. By no stretch. We have definitely not solved biology. Instead, what's really happened is the tools have allowed us to explore some types of questions, but they've created different types of research problems instead. So yes, we can sequence a lot of genomes very quickly and relatively cheaply. It's still not a trivial expense, but it's a far cry cheaper than it used to be. But a given genome is typically going to be megabases in length. And so that's a ton of information. And yeah, we have some computational methods to try and analyze what's going on, but in a lot of cases, our ability to generate data has greatly outpaced our ability to know what to do with that data. Along with that, although the techniques I've showed you tend to work fairly well with a lot of the conventional study organisms that biologists use, they don't always work as well as expected outside of those contexts. So for example, what I showed you for RNA interference, I've heard many stories from colleagues of trying to use that to study some gene in some type of animal, and they just can't get the RNA interference to work as they would expect it to. So, we're discovering the limits of these methods just as we're discovering a wide range of applications for them. And in addition to that, yes, this might let us figure out, what gene X does, but that still doesn't always provide us with the full picture for understanding how an organism functions. So just knowing something about an organism's genetics doesn't always tell us what's happening on the phenotypic level. And it might tell us what's happening in a particular tissue type, but not across the organism in general. So we still have to study different biological systems from multiple different angles.